Hello, everyone. Today, we'll be exploring the ICH Q5C Guideline on stability testing of biotechnological and biological products. This guideline is a cornerstone for ensuring the quality, safety, and efficacy of biotech products throughout their shelf life. My aim is to break down the guideline into practical insights you can apply in your professional context. The Q5C guideline was established by the International Conference on Harmonization, now known as ICH, to harmonize regulatory expectations across the US, Europe, and Japan. Biotech products, unlike small molecules, are extremely sensitive to environmental conditions like temperature, light, and oxidation. Even small changes can cause protein denaturation or loss of activity. This guideline ensures that companies generate reliable data, proving that their biologics remain stable under the conditions they will be stored, transported, and used. The scope of Q5C is quite specific. It applies to well-characterized proteins and polypeptides, such as cytokines, erythropoietins, monoclonal antibodies, growth hormones, and insulin. It explicitly excludes products like antibiotics, vitamins, blood, or whole cellular components. Why? Because the stability mechanisms differ significantly. For example, antibiotics degrade chemically, but proteins are much more vulnerable to physical changes like aggregation. Stability testing for biologics is not straightforward. Proteins rely on delicate non-covalent bonds to maintain their structure. If that structure changes, so does the biological activity. This means factors like temperature shifts, exposure to light, changes in ionic strength, or even mechanical stress can cause irreversible damage. That's why stability programs for biologics must be much more stringent compared to small molecules. Professionals in manufacturing, quality assurance, and regulatory affairs must understand these sensitivities when designing testing protocols. ICH Q5C emphasizes long-term, real-time, real-condition studies as the foundation for stability claims. At least 3 representative batches must be tested. Matrixing and bracketing strategies can be used to reduce workload, but only when scientifically justified. The use of advanced analytical methods, biochemical, immunochemical, and physicochemical, is required to detect subtle degradation. This approach ensures the data reflects how the product will actually behave in the market, not just in ideal lab conditions. One of the most important aspects is developing a stability indicating profile. No single test can give the complete picture, so manufacturers must combine multiple assays. Potency testing ensures the product retains biological activity. Purity testing checks for degradation products or aggregates. Molecular characterization ensures the protein hasn't fragmented, oxidized, or lost critical functional groups. Physical parameters like appearance, pH, and particulate matter are also monitored. Think of this profile as a safety net, catching any sign that the product is drifting away from its intended quality. Storage is critical. Most biologics require strictly defined refrigeration, typically 2 to 8 degrees Celsius. Deviations, even short ones, can cause irreversible damage. ICH recommends real-time studies at intended storage temperatures, accelerated and stress studies to simulate transport accidents or environmental exposure, special evaluation of container/closure systems, since proteins can adsorb onto glass, rubber, or plastic. And importantly, freeze-dried products must also be tested for stability after reconstitution, since that's when healthcare providers will actually use them. Testing frequency depends on the proposed shelf life. For short shelf life products, less than equal to 1 year monthly in the first 3 months, then quarterly. For longer shelf life products, greater than 1 year quarterly in year 1, biannually in year 2, and annually thereafter. Once enough real-world data is collected post-approval, testing frequency can sometimes be reduced. But the underlying principle is to prove stability continuously, not just at the beginning. Labeling plays a direct role in patient safety. ICH requires that labels clearly specify storage temperature ranges, whether the product must be protected from light or humidity, Instructions for reconstitution and use after reconstitution. This ensures that not only the manufacturer, but also healthcare professionals and patients handle the product properly. Inaccurate or vague labeling can lead to real-world product failures, even if the drug was perfectly stable in the lab. To summarize, ICH Q5C establishes a rigorous, science-based framework for proving the stability of biotechnological and biological products. It ensures patients receive drugs that are safe, effective, and of consistent quality throughout their shelf life. For professionals, the key takeaway is that stability is not just a regulatory checkbox. It's an integral part of product development, commercialization, and patient trust. Thank you. I'll be happy to take questions in comment box or discuss specific case examples from your areas of work.